Howdy all, Jack here. I'm going to do another 4-part video series on kind of functional neurology. So before the advent of microscope, or particularly the microscopes that allow us to look at living tissue, brains were kind of this black box of, we saw that we could put things into it and it would output something. So I'm going to be focusing on the putting things into it kind of element, and we might get into some of the functional bits that we know about that, but we're going to focus mostly on this input phase of neurology or neuroscience. So I'm going to give some background on the brain and our current conceptions of it in science and the evolutionary science of it. And then we're going to talk a little bit about the external senses, which I'm sure you're all very familiar with, because we think and talk a lot more about external senses than internal senses. But that's going to be this video. In the next couple of videos, we're going to talk about a couple different major internal senses. And then in the fourth video, we're going to talk about the sixth sense, or at least that's what the Buddhists tell us. And I'll let you ponder that for a little while, unless you already know, in which case you don't have to ponder, but you'll find out towards the end of the week. I think I'm going to try to bust these out before Sunday. Anyway, so some background on the brain. So the brain is essentially a system of coordinating movements and behaviors based on inputs. And those inputs are what we're going to be talking about, but those inputs are senses and whatnot, internal and external, and the outputs, the behaviors are muscle movement and eating and all of these sorts of things. So the earliest organisms were single-celled. They didn't need a brain or a nervous system to behave because they had a really simple system of, I'm just going to use flagella because there are different ways of doing it, but essentially the simplest method for a single cell to move is this spiral flagella. And it can turn two different ways, and it has a sense of, is there something I want in front of it, right? Is there food in front of me? And is there something aggressive behind me? Or, I guess that's also in front of me. So there's one kind of sense and there's one output. And the output can only go forward or tumble. If the flagella turns one way, the bacterium moves forward. And if it moves the other way, it moves chaotically until it finds a direction it wants to move forward, and then it moves that way. So with this sense, it says, do I want to go forward or do I not want to go forward? And if the answer to I want to go forward is no, or if the answer to I don't want to go forward is yes, then it tumbles. And as soon as the sense gets the other trigger of I do want to go forward or I don't not want to go forward, it starts going forward again. Fast forward a few million years to the first neural structures we see, and these tend to be in kind of flatworm sort of organisms that have a little more complicated. There's a whole bunch of cells all strung together. There's muscle tissue that helps it move. They wriggle through the water. And they have more complicated behaviors of what to eat, what to run away from, what to interact with. They're still very primitive seeming, but they are more complex than tumble or go straight. So this is the real origin of why we have neural structure. Then fast forward again, you've got lizards and things, and they have significantly more complicated than the flatworm, but they are yet to make another leap into the kind of mammalian complication. And with this jump towards to lizards is where you start to see kind of the rudimentary thoughts and habits and kind of these self-propagating behaviors, generally. And then you see they're more complicated in mammals. Interestingly enough, they're also more complicated in birds, despite the anatomically being very similar to lizards, and they don't seem to show the complexity of mammalian brains, but they do have more complex thought patterns, self-propagating kind of behaviors than lizards and whatnot seem to. I don't know why. But we are this kind of the development of the brain taken to its illogical conclusion, or to its logical conclusion, depending on who you ask. But we have this hugely incredible ability to take in data, arrange and rearrange information, remember things relevant to it, which all of those are functions of a brain. But we just take it to another extreme of why we do this, how we develop complicated languages to help us organize our thoughts and really take this brain thing to the next level. So I think that's a good overview on why we have brains. I'm not going to get into any of the theories on why humans have particularly interesting brains, because there's lots of different people saying this reason or that reason, and I don't really know. Or we have cool brains. If somebody comes up with a convincing theory, I'll be interested, but I don't have a real burning desire to know why we have interesting brains. We just do, so we gotta live with them. And so the next thing is the inputs that we talk and think the most about. So I won't go too in depth, but we have touch, right? We've got little pressure sensors and heat sensors all over our skin. We have taste. We've got a bunch of different chemical receptors on our tongue. We've got smell, which is more or less just really complicated taste. And it goes through a different nerve. Cranial nerves. Most senses have their own cranial nerves, with the exception of touch. So smell and taste are kind of related. They're essentially chemoreceptors in mucous membrane tissues that pick up the chemicals that you either breathe in or breathe out or run over your tongue. There's, of course, hearing, which you're doing to me right now. This is essentially a pressure wave from the world hits a floppy bit in your ear that transmits these pressure waves into a more usable pressure wave in a denser material, a liquid. And then you've got all these little hairs at different thicknesses of a tube that your brain knows how powerful this wave was by how far it gets down the tube. And that's a really oversimplified version, but that's essentially how hearing works. And then there's sight, which hopefully you're also looking at me. And that happens because light waves get refracted through a lens in your eye and get projected onto this essentially part of your brain. These light receptive neurons that coat the back of your eyeball, and they turn those individual little instances of light into a meaningful picture using the back of your brain. Let's see, I've talked about touch, smell, taste, hearing, and sight. 5 senses. So these could be further differentiated. Like I said, we've got 2 different, I think I said we had pressure and heat, but we've got 2 different types of pressure sensitive neurons and a heat sensitive neuron in the finger. So you've essentially got 3 senses that all sum to touch. You've got another 5 that sum to taste, about 1,000 different chemoreceptors in your nose. Again, you have essentially one type of neuron in the ear, but they're all tuned to a different tone, essentially. But that's a structure, not a neurological difference. And then there are 3 different color receptors, and then a dedicated light receptor in your eye. So that's another 4. So depending on how you cut senses, we could have 5, we could have, 1,013. But 5 senses works just well for most scenarios. I think I rambled a bit there at the end, but I hope you enjoyed my quick overview of external senses and why we have a brain. I'll be back tomorrow with the gut and the gut's influence on the brain and how that's one of the most important internal sensors we have. All right, see you later. Bye.